Welcome back to the deep dive. I am so excited for today's session because we are going to be talking about something that, well, it sounds like the setup to a really strange, really abstract joke, but it actually ends up being one of the most important cautionary tales in, I think, the history of modern science. We are going to talk about ghosts, we are going to talk about machines, and we are going to talk about a dead fish.
A very famous, very dead Atlantic salmon, to be precise.
It really is a legend, isn't it? I mean, if you hang around neuroscientists or statisticians long enough, someone is going to bring up the salmon.
It's inevitable.
But for the uninitiated, for you listeners who haven't spent your Friday nights, reading methodological critiques of functional magnetic resonance imaging, we have to set the scene. It's just too good to gloss over.
It really is. So picture it. It's 2009, and we've got a team of researchers, one of them a neuroscientist named Craig Bennett, and they're doing what is basically a calibration test. They're working with an fMRI scanner.
And that's functional magnetic resonance imaging.
Right. Those huge, incredibly loud donut-shaped machines, millions of dollars. They're used to map brain activity.
And the usual subject is, well, a living human. You put them in, you give them some tasks like tap your finger, look at this picture, and then you see which parts of the brain light up with blood flow.
Exactly.
Yeah.
But this day, the team just needed something to test the scanner's parameters on. Things like contrast, signal-to-noise ratios. They didn't actually need a living person. They just needed something with a complex biological structure.
So they went to the supermarket.
They went to the supermarket.
I love this detail. They didn't order some custom phantom from a medical supply company. They went to the fish counter.
They bought a mature Atlantic salmon. I think it was about 18 inches long, weighed 3.5 pounds, and it was undeniably deceased.
Right. This was not a fish that was merely resting.
No, or pining for the fjords, no. It was dinner.
So they take this dead fish, they wrap it up so it doesn't get the scanner messy, and they slide it into the bore of this multi-million-dollar machine. And they could have just, run a simple structural scan and called it a day, but they decided, maybe out of scientific thoroughness or maybe just a bit of lab whimsy, to run the full standard functional protocol, the exact same one they'd use on a human volunteer.
And this is where the paper's title comes from. Neural Correlates of Interspecies Perspective Taking in the Post-Mortem Atlantic Salmon. End quote. They actually showed the dead salmon a series of photographs, standard stuff from social psychology, pictures of humans in various social situations, happy, sad, surprised.
And they didn't show it the pictures, they talked to it.
They did. They read a standardized script to the salmon. They asked the fish to look at the humans in the photos and determine what emotion that person was experiencing. They asked a dead salmon to engage in perspective taking.
Which is an incredibly high-level cognitive function. I mean, I know some living humans who struggle with perspective taking, let alone a fish that's been on ice at the grocery store.
Precisely. So, on the surface, it's just this funny story about scientists having a bit of fun with their equipment. But the horror story, the scientific horror story, begins when they analyze the data.
Ah, yes.
They took the magnetic recordings from the scanner, all data representing changes in the fish's brain while it was looking at these pictures, and they ran it through the standard statistical software, the same software used by thousands of researchers all over the world.
And this is the punchline that really shook the entire field.
The software found something. It found activity. Specifically, when they looked at the results, there was a cluster of active voxels. Those are like 3D pixels in a brain scan, right in the salmon's brain cavity, and a few along its spinal column. The statistics suggested, with a standard level of confidence, that the dead salmon was actually thinking about the emotions of the humans in the photos.
Okay, let's just pause right there, because this is the absolute crux of everything we're talking about today. Biologically, we know for a fact that is impossible. The neurons were dead, there was no blood flow, no oxygen being metabolized. It was a hunk of meat.
Correct. There is zero plausible biological mechanism for that signal.
So if the biology is impossible, you have to look at the tool. Did the multi-million-dollar machine break? Was there, a ghost in the machine?
The machine was working perfectly, the magnets were fine, the sensors were recording. The problem wasn't the hardware, and it wasn't even the software, not in the sense of a bug or a glitch. The problem was the math, or maybe more accurately how we interpret the math. This whole study is the perfect, and I mean perfect, demonstration of a statistical hazard known as the multiple comparisons problem.
The multiple comparisons problem. That's our villain for this entire deep dive.
It's the monster in the closet for anyone, really, who works with large data sets. The authors of the salmon study, They weren't actually claiming the fish was psychic. They were publishing this to prove a point. They were showing that if you look for a pattern in enough places, if you ask enough questions of your data, you will eventually find a significant result purely by random chance.
It's the old idea that if you flip a coin enough times, you'll eventually get 10 heads in a row. And if you only report that one streak, you might mistakenly think the coin is rigged.
Exactly. And an fMRI scan is like flipping, 100,000 coins all at the same time. And that sets the stage for our entire discussion today. We're going to unpack the concept of the family-wise error rate, or FWER. It sounds like really dense jargon, but it's basically the gatekeeper of scientific truth. It's the only thing standing between us and a world where dead salmon are considered, I don't know, empathetic philosophers.
We have a lot to get through. We're going to define this error rate. We'll look at the classic ways researchers try to fix it, the so-called sledgehammer methods, and then we're going to get into the really sophisticated data-driven methods that involve, like, shuffling data thousands of times and building custom noise maps.
And we'll even touch on the philosophical side, because believe it or not, deciding what even counts as a family of tests is, well, it's more of a philosophical debate than a strict mathematical one.
But let's start at the very foundation. To really get why the fish looked like it was thinking, we have to understand how scientists decide that anything is true. We have to talk about the p-value.
The p-value. It is probably the single most used and most abused metric in all of science.
Right. Most people have probably heard of that magic number, 0.05. If your number is lower than 0.05, you pop the champagne, you publish the paper, you get tenure. If it's 0.06, you quietly file the results in a drawer and try again. But what does that 0.05 actually mean?
It is so widely misunderstood. A p-value is, at its heart, a probability. Specifically, it's the probability of getting a result, a test statistic as extreme as the one you observed, assuming that the null hypothesis is true.
Okay, that's the textbook definition. Now let's really unpack that last part. Assuming the null hypothesis is true, because that's the anchor for everything.
Right. In science, we always have to start from a position of skepticism. The null hypothesis is the boring stance. It's the default. It's the stance that says there is no effect. This drug is just a placebo. These two groups are identical. Or, in this case,
The salmon is dead.
the salmon is dead and is not thinking.
Yeah.
Right. So we assume that's true. Now, even in a totally dead salmon, there is noise. The scanner itself has thermal noise. Electrons are just bouncing around. There are tiny magnetic fluctuations happening all the time. So the p-value asks a very specific question. If we assume there is absolutely no real brain activity happening, what are the odds that all this random noise would just happen to arrange itself to look like a signal this strong?
So it's a measure of surprise. Like, how surprised should I be by this data if I assume that nothing is actually going on?
That's a fantastic way to put it. And the standard threshold for that surprise, what we call alpha, is usually set at 0.05. This means we're saying, I am willing to accept a 5% risk that I will be fooled. A 5% risk that I'll falsely conclude there's a real effect, when in reality, it was just a fluke of the noise.
And that's a type I error, a false positive, crying wolf when there's no wolf.
Exactly. And a 5% risk, I mean, that sounds pretty reasonable, doesn't it? If I told you there's a 95% chance you'd be safe crossing the street, you'd probably feel okay about it.
I mean, I'd prefer 100%, but sure, in the messy world of data, 95% confidence feels like a pretty solid standard.
And it is a solid standard, provided you are only doing one test. If I have one coin and I want to know if it's weighted, and I run one single experiment, a 5% error rate is acceptable. The problem, and the reason the salmon is so famous, is that modern science almost never involves just one test.
Which brings us perfectly to the jelly bean analogy. This is a classic example from the source material used to explain this exact problem.
It illustrates the point perfectly.
Good.
So imagine a scenario where a newspaper commissions a study to find out if jelly beans cause acne. That's the research question.
Very serious investigative journalism.
So the scientists go to the lab. Their null hypothesis is jelly beans do not cause acne. They get a group of people, they feed them jelly beans, they count the pimples, they run the statistical test. The p-value comes back at 0.50.
Which is way, way higher than 0.05. So, result, no link. Jelly beans are safe. End of story.
But then someone in the room says, hang on, wait a minute. Maybe it's not all jelly beans. Maybe it's the specific color of jelly bean that's the problem.
Ah, and so they break it down. They start testing the colors one by one.
Exactly. They test purple jelly beans. P-value is 0.20, no link. They test brown jelly beans. P-value is 0.15, no link. They test pink, blue, teal, yellow.
They just keep rolling the dice.
They keep rolling the dice. They test 20 different colors. Now let's assume, for the sake of reality, that jelly beans absolutely do not cause acne. The null hypothesis is true for every single color. But remember, for each one of those tests, we accepted a 5% risk of being fooled by randomness.
A 1 in 20 chance of getting a false positive.
Exactly. So if you roll a 20-sided die 20 times, what are the odds that you're going to roll a 1 at least once?
Intuitively, it feels like it's got to be pretty high.
It is. The math works out that the probability of getting at least one false positive across that family of 20 tests is about 64%.
Wow. Okay, that is a massive jump. We went from a 5% risk on a single test to a 64% risk of being wrong somewhere in the batch just by checking all the different colors.
You are now more likely than not to find a completely fake result. So in our analogy, the test for green jelly beans just happens, by pure random luck, to come back with a p-value of 0.04.
Which is below the magic 0.05 threshold. So the scientists write the paper, green jelly beans linked to acne, p 0.05.
And the other 19 tests that showed absolutely nothing, they get ignored, filed away. This is the multiple comparisons problem in a nutshell. You have conducted a family of tests. The error rate for any single test might be low, but the family-wise error rate, the chance that the whole family contains at least one lie, has completely exploded.
So family-wise error rate, or FWR, is basically the probability that at least one of your conclusions in a whole batch of tests is a false positive.
Correct. And we should probably distinguish this from something called the per-family error rate, or p-error. That's slightly different. P here is the expected number of errors. So if you did 100 tests at an alpha of 0.05, you'd expect to get 5 false positives. That's a count. FWER is a probability. It's the risk that your entire study, your entire set of claims, contains a fundamental flaw. And in a rigorous science, we really want to control that FWR. We want to be able to say the chance that any of these results you're reading is a fluke is kept below 5%.
Okay, so let's bring this back to the salmon. In the jelly bean example, we had 20 tests. How many tests, how many jelly beans are we talking about in a typical brain scan?
An fMRI scan divides the brain into those voxels we mentioned. Depending on the scanner's resolution, you could have anywhere from, say, 40,000 to maybe 130,000 individual voxels.
So instead of 20 jelly beans, we have 100,000 jelly beans.
Exactly. Now imagine rolling that 20-sided die 100,000 times. You are mathematically guaranteed to roll thousands and thousands of ones. You will find thousands of voxels that appear to be significant just by pure chance.
So the thinking signals in the dead salmon were just the green jelly beans of the brain scan.
Precisely. They were the statistical noise that just happened to cross that arbitrary 0.05 threshold because nobody had adjusted the threshold to account for the sheer number of tests they were running. If they hadn't corrected for it, they would have been forced to publish a paper claiming a dead fish has empathy.
And because they did correct for it, well, actually, the point of their paper was to show what happens when you don't. But I'm assuming once they applied the proper correction, the signal just vanished.
Yes, completely. Once the proper statistical corrections were applied, the activity was gone. The brain went back to being statistically dead, which of course matched the biological reality.
So how do we do that? If I'm a researcher and I have 20 jelly beans or 100,000 voxels, how do I stop myself from being fooled? What are the standard tools we use to fix this inflated error rate?
The most classical method, the one you'll find in every single Intro to Stats textbook, is the Bonferroni correction.
Bonferroni. It's named after Carlo Emilio Bonferroni. It sounds like a delicious type of pasta, but I get the feeling it's actually a bit of a buzzkill for researchers who are hoping for fame and fortune.
It is notoriously strict. A total buzzkill is a good way to put it. The logic, however, is beautifully, brutally simple. If you want your overall family-wise error rate to be 0.05, and you are conducting, let's say, 20 tests, you just take that 0.05 and you divide it by 20.
Okay, simple math. 0.05 divided by 20 is 0.0025.
Right, so all of a sudden, instead of needing a p-value less than 0.05 to claim a discovery, you now need a p-value less than 0.00025.
That sets the bar much, much higher for every single test.
It does. It effectively splits your total 5% risk budget among all the members of the family. If every individual test manages to meet that super strict threshold, then the mathematics of it guarantees that the risk of finding any false positive across the whole group is still just 0.05.
It works. It controls the error. It silences the noise.
It absolutely works. It is the sledgehammer approach, as you called it. If you use Bonferroni, you can be very, very confident that any positive results you find are real. But like with any sledgehammer, there's always collateral damage.
And I'm guessing the damage is that you might end up smashing some of the actual truth along with the lies?
Exactly. In statistics, there's always this trade-off. It's like a seesaw. If you push down really hard on the risk of a type I error, a false positive, you inevitably raise the risk of a type 2 error, a false negative.
A false negative. That's when there really is an effect to be found. Maybe green jelly beans do cause acne, but your study is too conservative and says they don't. You miss it.
Right. Suppose there's a real, but maybe subtle, link. Maybe the true p-value is 0.01. In a normal one-test world, that would be a significant finding. But under the harsh Bonferroni regime, with its new threshold of 0.0025, that 0.01 result gets rejected. You miss the discovery. You fail to find the cure for the disease.
And if we apply this back to the brain, 0.05 divided by 100,000 voxels.
You get a number with so many zeros after the decimal point that it becomes practically absurd. You would need a signal of astronomical strength to pass that test. If you used a standard Bonferroni correction on a whole brain analysis, you would almost certainly find nothing, even if the person was thinking very, very hard. You just wash out the baby with the bathwater.
So Bonferroni is safe, but it's maybe too safe. Are there more nuanced ways to handle this? Maybe a scalpel instead of a sledgehammer?
There are. There are a few variants. One is the IDAC correction, which is a little less conservative, but it assumes all the tests are independent, which is often not true. A much more common and powerful improvement is the Holm-Bonferroni method.
Holm-Bonferroni. Not related to Sherlock Holmes, I take it.
No relation, unfortunately, though it's a bit more clever. It was developed by a statistician named Stur Holm. It's what we call a sequential method.
How does that work? How is it different from just dividing by the total number?
With standard Bonferroni, every single test faces the same brutal, unchanging threshold. With Holmes' method, we treat the test differently based on how strong their initial result is. So imagine we have our 20 jelly bean tests. We get all our p-values back from the lab. The first step is to order them from the smallest p-value, the most significant one at the front of the line, to the largest, least significant one at the back.
Okay, so our green jelly bean test is at the front of the line because it had that p-value of 0.04.
Let's imagine it was even stronger, say 0.001 to make the example work. So green is at the front. Now we test that very first one, strongest result, against the strictest Bonferroni threshold. We take our alpha, 0.05, and divide it by the total number of tests, 20. If green fails to pass that strict bar, we just stop right there. We say nothing to see here. The whole family is non-significant. But, and here's the clever part, if green does pass that strict bar, we declare it a winner. It's a real finding. And then we move on to the second best result on our list.
And does that second one have to pass the same super strict test?
No, the bar gets a little bit lower, the hurdle gets easier to jump. For the second test in line, since we've already used up one test, we only have 19 tests left to worry about. So we divide our alpha by 19, not 20.
Ah, I see. So the penalty gets smaller. And if that second one passes, we move to the third and we divide by 18?
Exactly. It's a step-down procedure. As you confirm real effects, the statistical penalty for the remaining weaker tests gets progressively smaller. You are, in a way, rewarded for finding strong signals.
So this Holm-Bonferroni method gives you more statistical power. You're less likely to miss real, but maybe weaker, effects, but it still keeps that overall family-wise error rate locked down at 5%.
Yes. Mathematically, it provides the exact same strong control over the FWR as the classic Bonferroni, but with much better sensitivity. In modern science, there is almost no good reason to use standard Bonferroni when Holmes' method exists, except perhaps that the standard one is slightly easier to calculate in your head.
Okay, so those are the mathematical fixes. We adjust the threshold, we make the math smarter. But our research for this deep dive highlighted another way to handle the problem, a much more, let's call it a blunt alternative.
Yes, the blunt alternative is simply don't do so many tests in the first place.
Just stop asking so many questions of the data.
It sounds almost too obvious, but it cuts right to the core of scientific integrity and good planning. The problem of multiple comparisons often arises because researchers are, for lack of a better word, fishing. They're conducting what we call exploratory research, but they're trying to present it as confirmatory research.
Right, let's distinguish those two. It's like hunting versus gathering.
That's a good way to think about it. In proper confirmatory research, you should have a very specific pre-stated hypothesis before you even start. You say, based on prior theory, I have a strong reason to believe that only green jelly beans cause acne. You design the entire study to test only green jelly beans.
So you do one test, one comparison.
One test. Your alpha is 0.05. You don't need to divide it by anything because there's no family. There's just one child. Your statistics are valid.
But in exploratory research?
In exploratory research, you are honest and you admit that you don't really know what's going on. You say, I have no idea what causes acne, so I'm going to check everything. That's a perfectly valid and important way to do science, but you must pay the statistical price. You have to correct for the fact that you went on a fishing expedition.
And the problem is when people aren't honest about it.
Yes. There's a name for it, ARC-King. It stands for hypothesizing after the results are known. A researcher tests all 20 colors, finds by chance that green is significant, and then writes the introduction to the paper as if they had a deep, profound theory about the color green all along. They conveniently forget to mention the other 19 failed tests.
That's just cheating.
It's statistical cheating, yes. It completely invalidates the p-value. So the blunt solution is rigorous pre-planning. You pre-register your study, you write down in a public place exactly which tests you plan to run before you collect a single piece of data, and you stick to that plan. If you only plan for 3 comparisons, you only have to correct for 3. You don't let yourself get distracted by all the other noise and the other variables.
There's a specific trap that was mentioned in our sources related to this, something about a 2-track testing structure in factorial designs. This gets a little into the weeds of ANOVA, that's analysis of variance, but it seems important because it sounds like a lot of researchers fall for it.
Oh, it's extremely common, especially in psychology and the social sciences. So imagine a more complex study design. Maybe you're testing a new drug, and you have both men and women in your study, and you also have old people and young people. That's a factorial design. You have multiple questions you could ask. You could ask about simple effects like, does the drug work specifically for old men or specifically for young women? And you also have main effects like, does the drug work for people in general, averaging across age and gender?
Okay, so the temptation, I gather, is to run one big single omnibus test first.
Right. Researchers will often run a global test, an omnibus F test, that basically asks, is anything interesting happening anywhere in this entire data set? They treat this test as a kind of gatekeeper.
The logic being, if the gatekeeper test says, yes, something's going on, then they feel safe to go inside and look at all the specific groups without correcting.
Exactly. They think, the omnibus test was significant, so that gives me a license to hunt. I don't need to correct for multiple comparisons when I start testing old men versus young women. This is sometimes called the protected Fisher's LSD logic. But the sources we looked at argue this is often a fallacy.
Why is the gatekeeper unreliable? Why doesn't it offer that protection?
Because the gatekeeper is very easily distracted. The omnibus test only tells you that the global null is false, which just means that something is different somewhere. But let's say the drug has a massive, huge effect on old men, but absolutely zero effect on everyone else. The omnibus test will almost certainly be significant because that one huge effect in that one group.
So the gatekeeper opens the door?
The door swings wide open, the researcher walks in, they test the old men, bam, significant. Then they test the young women. Now, because they think they're protected, they're running that test at a raw, uncorrected 0.05 alpha. There's a 5% chance they'll find a false positive for the young women, just by luck.
And since the gatekeeper was already open, they report both findings. They might write a paper saying the drug works for everyone, when in reality it only worked for one group and the other was a statistical ghost.
Exactly. The protection offered by the omnibus test is weak. It doesn't strictly control the family-wise error rate for the specific hypotheses you actually care about. The expert advice here is pretty clear. Don't rely on a lazy gatekeeper. If you really care about what's happening in those specific groups, you need to plan for those specific comparisons from the start and correct for them directly.
Okay, so minimize the tests, pre-plan your analysis, don't rely on these vague global tests. That all makes sense for psychology experiments. But let's go back to our salmon.
Back to the brain.
You can't really minimize the number of tests in an fMRI scan. The whole point of the technique is to map the entire brain. You don't know ahead of time where the activity is going to be. That's why you're doing the scan. You have to look at all 100,000 voxels.
This is the domain of what we call mass univariate analysis. You are, by definition, doing a massive number of single tests. And as we already established, Bonferroni is just too harsh here. It would kill any real signal.
So what do we do if the sledgehammer is too heavy and minimizing the tests isn't an option? Do we just give up?
No. We turn to modern computing power. We turn to what are called data-driven approaches, and specifically to a beautiful technique called permutation testing.
I have to say, I really love this concept. It feels like we're hacking the very idea of statistics. Instead of using some generic formula from a dusty textbook, we use the data itself to build the rules of the game.
It's an incredibly brilliant and intuitive approach. Let's stick with a brain imaging example, but maybe use a human experiment to make it clearer. Suppose we're showing a person pictures of cats and then pictures of dogs, and we want to know if the brain reacts differently to cats versus dogs at any point in time.
Okay. So for every voxel in the brain or every time point in an EEG signal, we have a cat score and a dog score. We subtract one from the other to see the difference.
Right. And we get a map of differences across the whole brain. Some spots are high, some are low. But we still don't know if the high spots are real signals or just random noise. To find out, we use permutation. And the first step is to intentionally and completely destroy our own experiment.
Destroy it.
We take the labels in our data spreadsheet, the labels that say cat and dog, and we randomly shuffle them. We basically flip a coin for each trial or each subject. For subject 1, we might swap their data. We pretend their cat data is actually dog data, and their dog data is cat data. For subject 2, we leave it alone. We just mix it all up until the labels are completely meaningless.
So now we have a new data set where the labels cat and dog are just random assignments. There is absolutely no real biological truth left in this shuffled data.
Exactly. We have computationally sourced the null hypothesis to be true. We have created a parallel universe where we know for a fact that there's no difference between the conditions.
And then we run the exact same analysis again on this garbage data.
We run the exact same statistical analysis on this shuffled nonsense data, and we just see what happens. We see what kind of fake activity, what kind of signal pops up just from the random alignment of the noise.
And we do this just once.
Oh no, we do it thousands of times, maybe 5,000 times, maybe 10,000 times. We let the computer run all night. And in doing so, it builds up this massive library, this huge distribution of what random noise looks like for this specific data set.
That feels like the crucial difference, doesn't it? Bonferroni assumes some theoretical, perfect, bell-shaped curve of noise. Permutation testing uses the actual dirty, messy noise from your specific scanner with its specific quirks and your specific subjects.
Yes, that's the beauty of it. It respects the underlying structure of your data. If your machine have a weird 60 hertz hum, the shuffled data will have that same weird hum. You aren't making these broad theoretical assumptions that might be false. You are empirically building the null distribution from the ground up.
Okay, so we have 5,000 maps of pure random noise. How do we use those maps to save ourselves from the thinking salmon error?
There are two main strategies here, two main ways to use that distribution. The first, and maybe the most stringent, is called Tmax, permutation testing.
T-Max. Sounds like a brand of energy drink.
It could be. It stands for the maximum T statistic. So in each and every one of those 5,000 shuffled noise maps, we tell the computer to scan the entire brain and find the single highest peak of activity, the one loudest voxel in the entire brain map.
The king of the noise for that specific shuffle.
Exactly. We record that one maximum value for each shuffle. So at the end of the process, we have a list of 5,000 maximum noise peaks.
And that list gives us our new threshold.
It does. We sort that list from smallest to largest, and we look at the top 5%. That value becomes our new cutoff for significance. We're now saying, okay, for any result in our actual real experiment to be considered significant, its signal must be taller than 95% of the biggest, loudest noise peaks we ever saw in any of our random shuffles.
This is so clever. Because by comparing our real local voxel to the maximum noise found anywhere in the brain during the shuffles, we are effectively controlling the error rate for the entire family of voxels all at once.
Precisely. It provides what we call strong control of the family-wise error rate. And the beauty of the Tmax approach is its precision. If you find a voxel in your real data that is higher than that Tmax threshold, you can point to that specific coordinate in the brain and confidently say, this specific voxel is significant.
You can truly localize the effect. You can say the activity is right here at coordinates X, Y, and Z.
Yes, it gives you incredible spatial confidence. But as you might guess, there's always a trade-off. Since we are setting our bar based on the highest possible peak in the noise, the bar is still set pretty high.
It seems like it would favor sharp, tall, needle-like spikes of activity.
It does. But what if the real brain response isn't a sharp spike? What if it's a broad, gentle hill? A low hum of activity that's spread out over a large area of the cortex?
A smear of thought rather than a point of thought.
That's a great way to put it. The Tmax test might miss that smear entirely. The top of the hill isn't tall enough to beat the maximum noise spike from some other random part of the brain, so the whole effect gets ignored. For those kinds of signals, we use the second main method, cluster-based permutation testing.
The blob method.
The blob method, yes. In this approach, we don't look at single individual voxels anymore. Instead, we first apply some lower arbitrary threshold to the data, and then we look for groups of adjacent voxels that are all showing some activation above that line. We look for clusters, or blobs. And the statistic we measure isn't the height of the peak, but the mass of the entire cluster, which is basically the size of the cluster multiplied by the intensity of the signal within it.
So a really wide but low hill could actually have a bigger mass than a tiny but very sharp needle.
Exactly. So we run our thousands of permutations again. But this time, in each of our noise maps, we don't record the highest peak. We find the biggest random cluster that formed by chance, and we record its mass. We build a distribution of max blob sizes.
And if a blob in our real unshuffled data is bigger than 95% of the random noise blobs we found, we can call it significant.
We do. And this method has become incredibly popular in cognitive neuroscience because brain effects are often distributed and not point-like. It is much, much more sensitive to finding these weak, broad signals than Tmax is.
But, and the notes we have are very, very specific about this, there is a major critical caveat. This method only offers weak F-wear control.
This is a crucial distinction that, unfortunately, many researchers either don't understand or just gloss over. With cluster-based tests, the fundamental unit of significance is the cluster, not the individual voxel.
What does that actually mean in practice for a conclusion?
It means that if you find a significant blob of activity that covers the entire visual cortex, the only thing you can strictly statistically say is there is a significant effect somewhere within this blob.
You can't actually point to any specific spot inside the blob and say this spot is active?
No, you can't. You cannot confidently say this specific voxel at the very edge of the blob is active. And you certainly can't say the effect started at exactly 300 milliseconds just because the blob happens to start at that time point. The exact extent of a blob, its edges in space and time, are statistically fuzzy.
The analogy from our outline was about a weather report.
It's a perfect visual. The Tmax test is like reporting individual lightning strikes. You can say, lightning struck exactly at 12:05 p.m. at the corner of Main and First. It is precise and localizable. Cluster testing is like reporting a large storm system. You can say, there is a massive storm currently over New England. The storm itself is real. But you cannot be statistically sure that it is currently raining on one specific house in Boston just because that house is inside the storm cloud you drew on the map.
That is a subtle but absolutely huge difference in what you can claim. If a surgeon needs to know exactly where to cut, they need a Tmax result. If a psychologist just wants to know, does the brain in general notice the difference between a cat and a dog? Then a cluster test is probably fine.
Precisely. And the scientific error comes when people use the more sensitive cluster tests to find a blob, but then write their conclusions and make their figures as if they had used the more precise Tmax test. That's a statistical overreach.
So let's just pull back for a moment and directly compare these modern data-driven permutation methods to the classical Bonferroni-style stuff we started with. Why go through all the trouble of shuffling data 5,000 times on a supercomputer? Why not just use the simple math?
It really comes down to the fundamental nature of the data itself. Bonferroni and its variants like Holm are built on the assumption that every single test you're running is independent of all the others. It assumes voxel A has absolutely nothing to do with voxel B right next to it.
Which, for a biological organ like the brain, is a ridiculous assumption. The brain is the most interconnected network we know of. If one neuron fires, its neighbor is very likely to fire too.
Exactly. Biological data is heavily, heavily correlated. Bonferroni unfairly penalizes you for asking what are essentially redundant questions. It says, you just checked voxel A, and now you want to check voxel B, which is right next to it, and probably telling you the same thing. Too bad. That's a new test. I'm lowering your threshold again.
It's like it's double charging you for almost the same piece of information.
That's a great way to put it. Permutation tests, on the other hand, because they shuffle the whole data set together as an intact block, they implicitly capture and respect those correlations. They know that voxel A and voxel B are linked because they always stay linked in all of the random shuffles. So the permutation test doesn't penalize you as harshly for the sheer number of voxels. It offers a much, much better balance of statistical power and rigor for complex correlated data like you'd find in neuroscience.
So for the jelly bean example, where the different colors are distinct categories and probably are independent, a Holm Bonferroni correction might be perfectly fine. But for a brain map or a mat of gene expression, permutation is really the gold standard.
Generally, yes. It is considered the most honest and statistically sound way to handle high-dimensional biological data.
I want to pivot now to the final and maybe the most difficult segment of our discussion. We've talked about the math, the shuffles, the salmon, but there's this deeper, almost philosophical problem lurking underneath all of this. We kept saying family-wise error rate, but what exactly is a family?
That is the million-dollar question. And believe it or not, there is no single objective mathematical answer to it. It is ultimately a subjective conceptual choice that the
Huh.
researcher has to make and justify.
Our sources get into a real debate about this. How big is a family? Is a family just the tests you report in one figure in your paper? Is all the tests in the entire paper? Is all the tests a scientist does in their entire life?
That last one is sometimes brought up as the lifetime error rate, and it's really a reductio ad absurdum. If you had to correct for every single hypothesis you ever tested in your whole career, by the time you retired, your p-value threshold would be so infinitesimally small you could never publish anything. You'd be statistically paralyzed.
So clearly, lifetime is too broad. But one paper seems tricky too. What if I write a paper that has two totally different experiments in it? Experiment 1 tests short-term memory, and experiment 2 tests visual perception. Should I really be grouping them together and making the penalty for one dependent on the other?
The general consensus from the literature, and specifically from arguments by statisticians like Rubin and Garcia Perez, is that a family should be defined by the logical structure of the scientific inference you are trying to make.
Can you give me a concrete example of that?
Sure. Think of a doctor. A patient comes into the emergency room with a set of vague symptoms. The doctor orders a large panel of 10 different blood tests to try and diagnose this one patient.
Okay.
The doctor's fundamental question is this patient sick? If any one of those 10 tests comes back positive, the conclusion is, yes, this patient is sick and needs treatment.
So the tests are all working together to answer one single overarching question.
Right. The logic is a disjunction, a logical OR. If test 1 is positive, or test 2 is positive, or test 3, since any single positive result leads to the same overall conclusion, the patient is sick, those 10 tests absolutely form a family. You must correct for them. If you don't, you run a huge risk of telling a perfectly healthy person they have a terrible disease, just because one of the 10 tests flickered by chance.
That makes perfect sense. The family is defined by the claim.
Now, imagine the same doctor sees patient A and runs a test. Then, 10 minutes later, a completely different person, patient B, comes in, and the doctor runs a test on them. Does the doctor need to correct patient B's test results based on the fact that they just tested patient A?
No, that would be completely absurd. They're different people, different problems, independent events.
Exactly. Even though it's the same doctor and the same lab machine, the scientific questions are distinct and conceptually separate. Is patient A sick and is patient B sick? Are not logically linked. They're not part of the same inferential family.
So in our research, we have to ask ourselves, are my different hypotheses like one patient who is getting 10 different tests, or are they like 10 different patients?
Yes. If you're a pharmaceutical company testing a new drug and you want to make the broad claim this drug is safe, and you test for 50 different potential side effects.
You better correct for all 50 tests. Because if it causes even one of those side effects, your global claim that it is safe is false. That's a family. But if your paper is more exploratory and you're just claiming, I found 50 interesting separate things about the brain,
Then maybe you can treat them as conceptually separate discoveries, or you might use a less strict type of correction, like the false discovery rate, or FDR, which we haven't touched on much. That method doesn't control the chance of making even one error, but instead tries to control the overall proportion of errors among your discoveries. But for rigorous confirmatory science, you need to define your family based on the claims you make.
it's just fascinating to me that statistics, which we think of as being so objective and black and white, ultimately comes down to a human judgment call about intent and the logical structure of an argument.
It absolutely does. The math of the Bonferroni correction or the T-max permutation test is constant. It's just arithmetic. But the decision of when and how to apply it requires scientific judgment and philosophical clarity. You have to really understand the logic of your own research questions.
This has been a really fascinating journey. We started with a dead salmon, engaging in high-level social perspective taking, which turned out to be nothing more than a ghost in the machine of uncorrected statistics. We walked through the brutal sledgehammer of Bonferroni, the elegant computational shuffling of permutation tests, and we landed on the deep philosophy of how we even define truth in the first place.
It really just highlights that data doesn't speak for itself.
Mhm.
We have to speak for it, and the language we use to do that is statistics. And if our grammar is wrong, if we failed to correct for our multiple comparisons, we end up speaking complete nonsense. We end up with psychic fish.
And in this era of big data, where we have more fossils, more genes, more social media posts, and more variables than ever before, this issue is more critical than it has ever been.
Absolutely. The significant finding that you read about is only as good and only as trustworthy as the correction method that was applied to it. So when you see a headline or you read a paper, you should always be asking, how many jelly beans did they check before they found the green one?
So as we sign off, here's a final thought to leave you with. We spend so much of our time and energy trying to filter out the noise. We build these massive statistical walls, Bonferroni, Tmax, pre-registration, all to keep the false positives out. We are, and should be, terrified of the scientific embarrassment of the dead salmon.
But we also have to remember the cost of building those walls. Every correction we apply, every time we make our threshold stricter, we make it just a little bit harder to see the subtle, quiet, real truths that might be hiding just below that line.
The eternal trade-off between cynicism and discovery. Are we missing the next big breakthrough because our statistical methods are too conservative, because we're too afraid of being wrong?
Or are we flooded with fake news and unreproducible findings because we aren't afraid enough? The answer, I think, is probably somewhere in the shuffle.
Thanks for diving in with us.
It was a pleasure.
See you next time.